What's up, TUMNAD lectures? In this video, we're going to talk about what we call the neurophysiology. But please, if it is the very first time, please make sure you subscribe. You turn on notification bell so that whenever I load the video, you become the very first person to watch. So let's go ahead and get started and look at what we call the neuron physiology. During this study, you're going to be seeing this structure of the neuron, and this neuron is going to be part of what we call the multipolar neuron. And this is going to be what we call the motor neuron and continues to be among the biggest percentage of what we call of the nervous tissue. Okay, now we are going to be studying this motor neuron. We see the different parts and then the function of those parts. We shall look at the axonal transport and some little bit of what we call the graded potential. Okay, now we are going to first look at what we call the anatomy of these parts and how they are called, and then later we come up with what we call the functions. Okay, now, when you see these extensions which are going to be coming from this bulbar structure, how do we call these kind of extensions? These extensions are what we call the dendrites. Okay, are what we call the dendrites. Okay, there are going to be a lot of this kind of the dendrites which are going to be coming off of this kind of bulbar structure. Okay, so now how do we call this kind of round structure? Okay, this whole structure. The whole of this structure is what we call the cell body. Okay, is what we call the cell body. But you may find some other books or some other authors calling it the soma. Okay, you may find some other authors calling it the soma. Okay, now inside this cell body, you're going to be having different organelles. You're going to be seeing these organelles inside here. Like this is going to be the nucleus, housing what we call the DNA. You're going to be having these kind of structures, having the ribosomes, which you may call the rough endoplasmic reticulum, which can also be called the Niso bodies. Okay, you may find some other terms like other books saying what we call the Niso bodies. The same as what we call this structure, what we call the endoplasmic reticulum, located in the cytoplasm of the neuron. Then we have also what we call the powerhouse of the cell, which is the mitochondria. Then we have the packaging center, which we call the Golgi apparatus, and then very many other structures are going to be located within this cell. Okay, now how do we call, you see now, like this cell body, it has this area where it is going to start to narrow down. Okay, it's going to start to narrow down before it transitions into this tube. How do we call this structure? That the narrowing point of the cell body, we call this one as the axon. We call it as the axon hillock. Okay, we call it the axon hillock. Okay, now how do we call the whole of this tube which is going up to this side? The whole of this tube, is what we call the axon. Okay, this tube is what we call the axon. Okay, now how about we have these cells which are going to be residing in this structure? How do we call these kind of cells? These cells are what we call the Schwann cell. Okay, are what we call the Schwann cells. Okay, and then now how about the whole of this structure which is going to be wrapping around this kind of a neuron? How do we call this kind of structure which is going to be more of a fatty substance, making this axon to look like white? It is white in nature, it is a fatty substance. This structure is what we call the myelin, fish. Okay, is what we call the myelin fish. Okay, now how about this end, which is going to be more of the bulbous, more dilated structure at the terminal of this axon? We call this structure as the axon terminal. Okay, is what we call the axon, terminal. Okay, is what we call the axon terminal. Now, looking at these kind of the myelin fish, which I've said the fatty substance, we have spaces which are going to be in between these kind of the myelin fish. What is going to be these spaces? How do we call these spaces generally? These spaces are what we call the nodes, of Ranvier. Okay, they are called the nodes of Ranvier. Okay, we shall see what these nodes of Ranvier, what they do. Okay, so now having looked at now the diagrammatic illustration by and all the parts that make up this kind of the motor neuron, let's go ahead and see what do these parts play generally. Okay, in the part of what we call the physiology. Okay, now we want to see what do dendrites do. Okay, These dendrites, they are going to be the receptors. They are going to be the receivers of electrical signals that is going to be coming to this cell body. Okay, they are going to be now receivers of the signals. Okay, the dendrites are going to be, they are going to be receivers. They are going to be receivers of signals. Okay, they are going to be receivers of the signals. Okay, what else? What is going to be the role of the dendrites? These dendrites, they play a role in the generation of what we call the graded, potential. Okay, what we call the graded potential. Now, we need to first know what graded potential is. Graded potential is going to be that localized potential difference, which is going to be on the dendrites and then also on the cell body membrane. Okay, so now that localized potential difference that's going to be generated on the cell body and also the dendrites is what we call the graded potential. Okay, so now let's even make it more clear physiologically. You know, we are going to be having a lot of the neurons. We're going to be having a lot of neurons which are going to be coming to synapse with this neuron. Okay, so now let me say like this neuron is going to be now our motor neuron. Okay, this is going to be our motor neuron. Okay, this one is going to be now our motor neuron. We have thousands of the neurons which are going to be synapsing with this kind of the neuron. Okay, so now when we have this kind of the neurons which are coming to synapse with this kind of the axon and also on the cell body, now we are going to cut a very small section. We're going to cut a very small section on the dendrites. We zoom it and see exactly how are these presynaptic neurons, how they are going to be stimulating this kind of postsynaptic neuron. Okay, so now if we cut up this structure and zoom it here, we are going to be seeing something like this. Okay, we are going to be seeing something. We are going to be seeing something like this. So now, when we zoom up this area, we are going to be having a lot of these proteins. Okay, we are going to be having a lot of these proteins. Okay, we are going to be having a lot of these proteins. Okay, so now, how do we call these kind of proteins? These proteins, they're going to be having a very unique receptor. Okay, they're going to be having a very, very, very important receptor where different chemicals are going to bind. So now these kind of the channel protein, these iron proteins, they are what we call the ligand. They are what we call the ligand. They are what we call the ligand. They are what we call the ligand gated. They are what we call the ligand gated ion channels. Okay, the ligand gated ion channels. We want to see when this kind of the motor neuron, which we call the presynaptic neuron, when it comes to synapse with this kind of the dendrite or the cell body, it is going to be releasing such kind of neurotransmitters. Okay, so now let's say like for example this kind of the neuron is going to be coming and it's going to be releasing a neurotransmitter on this kind of this ligand gated ion channel. Okay, so now we want to say these kind of neurotransmitters can be stimulatory or they can be inhibitory. So now I want to see that for example this one is going to be now an excitatory neurotransmitter. For example, we have excitatory neurotransmitter like we have what we call like glutamate. Okay, like glutamate. So now if this kind of neurotransmitter comes and binds on this receptor, it is going to cause this kind of the ligand ion channel to open up. When it opens up, guess what happens? We're going to be having entrance of different cations. Primarily, we're going to be having entrance of what we call sodium ions. So you're going to be having a lot of sodium ions coming inside here. So now, when there are a lot of sodium that enters into this kind of the membrane, it is going to be making that kind of membrane to become more positive, or you may even call it less negative. We call that one as that areas we call it depolarization. Okay, we call it depolarization. And then another good term we can use here is what we call EPSP. Okay, excitatory postsynaptic potentials. Okay, then you're going to be having also another kind of the neuron which is going to be releasing another neurotransmitter. We want to say that this neurotransmitter is an inhibitory one. And then, of course, an inhibitory neurotransmitter, we say, for example, let us use what we call GABA. So now when this neurotransmitter comes and binds on this kind of the receptor, it is going to cause this ligand ion channel to open like this. When this ion channel opens, you're going to be having now entrance of what we call the anions. Primarily, you're going to be having what we call the entrance of what we call chloride. And of course, chloride is going to be negatively charged. But what we know that inside of this membrane is already having high levels of negative ions. What does that mean? It means you're going to be making this area to become even more and more and more negative. Okay? How do we call whenever a cell membrane becomes even more and more negative? We call that one as a hyperpolarization. Okay? We call that one as hyperpolarization. Okay? And then another good term we can use here is what we call IPSPS. Okay, so now we say that inhibitory postsynaptic potentials is going to be generated. So now these two terms collectively are what we are going to be calling what we call the graded potential. Okay, the graded potentials. Okay, so now the graded potentials, they are going to be generated by both the membrane of the cell body and also what we call the dendrites. Okay, so now they are all going to be generators of what we call the graded potentials. Okay, we have said that the graded potential is going to be that local potential difference which is going to be generated on the cell membrane of the cell body and then also on what we call the dendrites. Okay, so now we have seen that how the dendrites are going to be generating what we call the graded potential. Again, we have said that even the cell body has the ability to generate what we call graded potential. Okay, graded potential. So the cell body can also generate what we call the graded potential. What else do you think is going to be played by what we call the cell body? You know, the cell body, it does again another important and beautiful role. It plays a role of what we call protein synthesis. Okay, it plays a role of what we call protein synthesis. Okay, of what we call protein synthesis. You know, when we are talking about the protein synthesis, this is what's going to be happening. You know, for a particular protein to be made, we need to have an individual kind of gene which has to be expressed. And where do we have our genes? We have this kind of the nucleus which is going to be residing in what we call the cell body. So now, if this kind of the gene is going to be now expressed, we are going to be having now what we call transcription, of which transcription is going to be leading to formation of what we call messenger RNA. You know, whenever you want to make a particular protein, you need to first, the genes have to undergo what we call transcription, which leads into the formation of what we call messenger RNA. Now, messenger RNA has the ability to leave the nucleus through the nuclear pore and comes out through the cytoplasm, where it goes into these kind of protein machineries. How do we call these protein machineries? The whole of this structure we said is what we call the rough endoplasmic reticulum, or you may call it the initial body. So now this kind of the messenger RNA is going to get translated by these ribosomes to make a protein which we want. Say, for example, this protein, let's say that, for example, this is going to be now our protein, which is going to be in this vesicle. Now, we are going to take this kind of protein into this kind of the structure, which we are calling the Golgi apparatus, for proper packaging. So now this kind of the protein is going to undergo the packaging process from the Golgi apparatus, and then it will bud off. Okay, it will bud off, and then we shall have now our fully packaged vesicle, and this vesicle is going to be containing different kind of proteins. What are kind of the proteins which are going to be residing inside this kind of the vesicle? This protein, they can be enzymes. Okay, this protein, this kind of vesicle can be containing what we call enzymes. We may have a lot of enzymes inside there. We may have even what we call the neurotransmitters. Okay, the neurotransmitters. We may even have what we call structural protein. Like, for example, structural proteins that leads into the formation of some of these structural cytoskeletons like the microtubule. We may even have what we call membrane proteins. We may even have what we call membrane proteins. Okay, membrane proteins. So you realize that the cell body having this kind of genetic material is going to be aiding in what we call protein synthesis for this kind of proteins, as we have just listed here in these vesicles. Okay. Now, that is for the case of the cell body. Now, coming on the axon, the primary role of the axon generally is going to be what we call conduction of action potentials. Okay, it's going to be what we call conduction of what we call action potentials. Okay, conduction of what we call action potentials. That is general primary role of what we call the axon. There is another role which is also not so much special, but is also very important, and this one is what we call axonal. Transport. Okay, is what we call axonal transport. We shall look later and see what axonal transport means. Okay. Now, coming on this strand cell, these strand cells primarily they are going to be responsible for production of what we call the myelin phase. Okay, they are going to be responsible for what we call producing what we call the myelin phase. That's going to be their main role. This one cell, they're producing the myelin phase, which is only kind of the fatty substance which is going to be produced by myelin phase in the peripheral nervous system. In the central nervous system, we have those kind of special cells which we call the oligodendrocytes, which does the same role as H1 cells in the peripheral nervous system. What's the role of the myelin phase? This myelin phase is going to be insulating the axon. Okay, they play a role of what we call insulation. Okay, they are playing a role of what we call insulation. And then also, this kind of the insulation is going to be aiding in faster movement of the electrical impulses. For that reason, they're going to be aiding in what we call faster movement. What we call faster movement of action potentials. Okay, what else? Don't you think these guys, they are also going to be protecting these axons? Yes, they are doing a role of protection. Okay, so they are going to be doing a role of what we call protection. Okay, that is for the case of the myelin phase. The myelin phase is going to be that kind of the fatty substance that's going to be doing the role of insulation, faster movement of the action potentials, and also doing a role of protection. Okay, how about this bulbous structure? You know, this kind of the bulbous structure is going to be containing different kind of vesicles. So for that reason, it can play a role of storage of these vesicles. Okay, storage and then release of the different neurotransmitters. Okay, so now this axon terminal is going to play a role of what we call storage, storage and release of neurotransmitters. Okay, storage and release of neurotransmitters. What else do you think is going to be played by this kind of the axon terminal? You know, this axon terminal, when electrical impulse crosses the neurotransmitters, we do not need them to remain in what we call the synaptic cleft. Some of these structures here, we have what we call these kind of the proteins that have the ability to reuptake some of the neurotransmitters or from what we call the synaptic cleft. So for that reason, the axon terminal can play a role of what we call reuptake. Okay, they can play a role of what we call reuptake of some of the neurotransmitters or from what we call the synaptic cleft. Okay. Now, coming from what we call the axon hillock, the axon hillock is going to be playing a role of what we call initiating the axon potential. Okay, it's going to be playing a role. It's going to play a role of what we call initiating. It's going to play a role of what we call initiating action. Initiating what we call action potential. Okay, initiating what we call action potential. You know, it is for the electrical impulse to flow down the axon. The generating point of the axon potential is going to be the axon hillock. You know what? It has a high concentration of sodium or voltage-gated sodium channels. For that reason, this is going to be a deciding point of whether a neuron is going to fire or not. Okay, so now the axon hillock is also very important. How about these nodes we are calling the nodes of Ranvier, these gaps? You know, when an action potential is generated, it's going to be jumping from one node of Ranvier to the other, from one node of Ranvier to the other. And that kind of the jumping of the action potential from one node to the other, we call it saltatory conduction. We call it saltatory. Okay, saltatory conduction. Okay, we call it saltatory conduction. So the nodes of Ranvier are going to be aiding in what we call saltatory conduction, and even also a regeneration of what we call the axon potentials. Okay, so now having looked at all now these parts with the roles which they play, let's go ahead and see what we call axonal transport, because this is super very important to know what we call axonal transport. Okay, I feel like you should know what we call axonal transport. Okay, so now what is going to be axonal transport? What is generally axonal transport? Axonal transport is that kind of transport of different substances from the cell body down to the axon terminal. And also, some of the substances need to move from the axon terminal back into what we call the cell body. So we want to see how is that one going to be happening within this axon, okay? You see this structure is going to be in the axon. This structure is going to be part of what we call cytoskeleton. And this structure is what we call the microtubule, okay? Now, this microtubule is going to be having these kind of transport proteins. You see these kind of the airplanes, they are what we call the transport proteins. These transport proteins, they are what we call the motor proteins. They are called what? They are what we call the motor, they are what we call the motor proteins. Okay, they are what we call the motor proteins. Okay, now we have a variety of them. We have these kind of motor proteins which are going to be transporting different substances from the cell body to the axon terminal, and we have those kind of the motor proteins that are going to be transporting substances from the axon terminal back to the cell body. So now, starting with this kind of the airplane which are facing the other way, these kind of the proteins which are going to be facing the other way, they are going to be like this. Okay, they are going to be like this. They are going to be like this. You see this kind of these proteins? These proteins, they are taking substances from this area going to the axon terminal. How do we call this protein? This protein is what we call kinesin. Okay, is what we call kinesin. And this kinesin, they are going to be transporting substances from cell body. They are transporting substances from cell body to what we call axon terminal. Okay, what we call axon terminal. Okay, so now this kind of movement of substances from the cell body to the axon terminal, this is what we call anterograde axonal transport. What we call anterograde axonal transport. Okay, so now what are some of these substances that need to move from the cell body to the axon terminal? We have this guy, the first one, which we call this structure is what we call the vesicles. Okay, what we call different vesicles containing different kind of proteins. Okay, we may also need some of the energetic materials here that are going to be providing the energy here in this process of what we call transmission. So we may need also the powerhouse, which we are calling the mitochondria. Okay, we may also need what we call mitochondria. Okay, mitochondria. Okay, so we may even need some other kind of structures that are going to be moving from the cell body down the axon terminal. But we have said that this kind of transport is what we call anterograde transport. Okay, so now let's look at again these motor proteins which are going to be moving things from this side to this side. Okay, and this kind of the airplane is going to be shaded. It's going to be shaded red. Okay, it's going to be shaded. It's going to be shaded red. Of course, see, this kind of an airplane, they are going to be transporting different substances. Now, this kind of the airplane or this kind of the motor protein that are moving things from the axon terminal to the cell body, they are what we call the dynein. Okay, they are what we call the dynein. Okay, and because they are moving things from the axon terminal to the cell body, we call that kind of transport as the retrograde. What we call retrograde axonal transport. Okay, now what are some of those structures that need to be transported from the axon terminal back to the cell body? We have a couple of them. For example, We may have some kind of the damage on the axon terminal, and we want to tell the cell body nucleus to generate more of the membrane proteins to come and do more of what we call repairs. So we may have what we call the neuron growth factors need to be transported by the dynein to the cell body to tell that we need more of the membrane protein to come and repair some of these areas which are damaged. Okay, we also have what we call infections. Okay, what we call the viruses. You know, we have different viruses that are going to be passing through the axon terminal, and they are transported to the cell body to use the genetic material for their replication. We have a couple of them. We have what we call, the first one is what we call the poliovirus. We have what we call the varicella zoster virus. We have what we call also the tetanus, All these viruses, they can pass through this kind of axonal transport, or what we call retrograde axonal transport, to come to the cell body. You know why they are coming to the cell body? It's because they want to use the genetic material inside here, this nucleus, to make their own copies. Okay? For example, this kind of the varicella zoster. You see, this kind of varicella zoster, when it moves to this area and it accesses the genetic material inside here, it's going to make its own copies, and these copies are going to be stay dormant for even a couple of years. But if you have any immune deficiency, or the immunity goes low, these kind of the particles of what you call the varicella zoster virus, through what we call the kinesin, they can come and then come out from the axon terminal, and they start damaging the skin. Okay, and this is what we call the herpes zoster, or even what we call the shingles. Okay, and then start damaging what we call the axon terminal. Okay, so now for the case of retrograde transport, these are the key areas that, and there are very many other things. Maybe, for example, even the mitochondria, its time of going has reached, it can also be taken back to the cell body. Okay, it is also such a beautiful thing. Okay, so now having looked at what we call the axonal transport, we have retrograde and then anterograde type of axonal transport. So now briefly, let's have a look at and see how these axonal terminals, how are they going to be releasing the neurotransmitters into the synaptic cleft? It is worth to be known. Okay, now we are going to be having this kind of vesicle here. You see this kind of vesicle here. This vesicle is going to be containing the neurotransmitters. We need to know how these neurotransmitters are going to be crossing this area into the synapse. You know what happens is come and study what we call the axon potentials. But of course, we want to say that if an axon potential arrives at the axon terminal, there are a lot of physiological processes which are going to be happening. Okay, when an axon potential reaches the axon terminal, we have these channels here. They are called they are called voltage gated calcium channels. So now these channels are also very important. When an axon potential reaches here, these kind of calcium ions are going to be start entering inside here. Now, what happens when calcium enters the axon terminal? You see, this vesicle is going to be containing what we call some kind of the proteins, and even also this presynaptic membrane is also going to be containing some kind of proteins. You know how these proteins are called? These proteins are what we call snare proteins. Okay, they are what we call snare proteins. Okay, these snare proteins are also very, very, very important. We have those kind of the proteins which are going to be on the vesicles, and also have those kind of snare proteins which are going to be on the presynaptic membrane. So now, I want to first cut the whole of this area. We zoom it and see how these kind of the proteins are going to be arranged on the vesicles and then also on the presynaptic membrane. So now, if this is our vesicle containing different neurotransmitters, we have this kind of the protein which is going to be there, and this kind of the protein which is going to be part of the snare protein is what we call synaptobrevin. We also have this kind of the proteins. This protein is going to be what we call synaptotagmin. Okay, synaptotagmin. And this protein is not going to be part of the vicinals, and this is going to be more of the calcium sensor. Okay, this synaptotagmin is going to be a calcium sensor that's going to be triggering the process of what we call exocytosis. Okay, so now these proteins are all characteristic. This kind of this synaptobrevin is going to be part of what we call vicinals. Okay, it's going to be part of the V snares. So now bringing this membrane here, this membrane, this presynaptic membrane is going to be having different kind of the proteins. There's this protein you may call it SNAP, you may call it SNAP 25, and then this protein down here, you may call it syntaxin. Okay, you may call it syntaxin. And collectively, these proteins, they are what we call T snares. Okay, they are what we call T snares. Okay, meaning that snare proteins, we have what we call T snares and also have what we call V snares. But synaptotagmin is not directly involved in what we call, is not part of what we call the V snares. They are what we call calcium sensors. Okay, synaptotagmin is what we call calcium sensors. Okay, they are what we call calcium sensors. Now, what happened is this synaptobrevin and then syntaxin and then SNAP25, they are going to be making a complex whereby the synaptotagmin will come and put this kind of the calcium. And when this happens, it is going to be leading to the pulling of this kind of the vesicle to come and fuse with this what we call the presynaptic membrane. This is what's going to be happening. So now this vesicle is going to come and fuse. It's going to come and fuse with the presynaptic membrane, and then eventually it's going to be powering the neurotransmitters in what we call the synaptic cleft. Okay, so now this is how the neurotransmitters are going to be moving into the synaptic cleft. And how do we call this process? This process is what we call exocytosis. And the trigger effect for this process is going to be what we call synaptotagmin, which is going to be a calcium sensor. It is such a beautiful thing. But now when these neurotransmitters have done their role, electrical signal, because here we're going to be having electrical signals, they are converted into chemical signals, and then finally they are going to be converted back again into electrical signals as electrical impulse crosses the synapse. But again, these neurotransmitters, some of them after the impulse has gone, they need to be taken back into what we call the axon terminal, or they are going to be broken down by different types of enzymes, or they are going even to be degraded by other kind of the mechanisms. These are going to be now what we call reuptake proteins. They are going to be taking some of these neurotransmitters back into the presynaptic terminal, what we call the axon terminal, maybe for more of the repackaging, maybe for use in case another axon potential comes and wants to cross the synapse. Okay. That really covers what we call part of what we call the neurophysiology. We have just talked about the parts, the function, the graded potential, and some little part of what we call the axonal transport. We shall later come and dive into what we call the axon potential. You're going to be having another full video of what we call the axon potentials. We shall again see what we call the resting membrane potentials. We see exactly how are they going to be connected with this kind of the neuron. Okay, I'm sure you have benefited from this video and you think whatever I've talked is making sense, is adding to you, you are really benefiting. Please make sure you like the video. You can comment in the comment section. Make sure you share this video to the people whom you think this video is making sense. And finally, make sure you subscribe. Thank you very much for watching. It has been TUMNAD from the beginning up to end. As always, until we meet next time.